Okay, so why don't we get started? Thank you guys for coming. This is group number 24, and they are presenting nanoparticles as nanoparticles for drug delivery.
So I'm Mariah, this is Issa, Shannon, and Julie, and together we make up Dendroline. Our mission statement as a company is that we strive to improve the efficacy of existing technologies with innovative and specified solutions utilizing unique nanoparticle characteristics.
So our goal is to use something called the dendritic polymer to help deliver the drug to the tumor site. The picture on the top you see is the structure of a dendritic polymer. So it has three main parts: a core, first generation, and nth generation, so as big as we need it to be to get to our required size, which we hope to be 100 nanometers, and the terminal functional group. For us, we're using something specifically called the dendritic polyglycerol, and that just means that our core is a glycidol ring that's being broken open. So these can be synthesized through two different methods, convergent and divergent synthesis. So we'll be using divergent synthesis, which means that it starts at a 0 and we'll build it outwards until it's this tree-like structure. So in between kind of all the branches is where the drug will be able to sit. So this is just a little bit more about the dendritic polyglycerol. We intend to distribute doxorubicin, which is an already FDA-approved drug to target the cancer cells. So on the right, the doxorubicin in DNA and the chemical structure are going to be attached where the terminal OH is here, so at the outside of the dendritic polymer. And once the polymer has been synthesized to about 100 nanometers, we're going to add something called a pH-sensitive linker, and that's what's going to help us get the polymer to the location. So cancer cells have very acidic extracellular environments, so that pH-sensitive linker actually allows the dendritic polyglycerol to essentially disintegrate and release the doxorubicin in an acidic environment. And lastly, this is just some research that we came across that proves the stability or instability of the pH-sensitive linker. So a study was done with the one that we intend to use, and this was the data that they released. So there was a 25-hour half-life without the pH-sensitive linker, and a 2-hour half-life in between 4 and 6 pH. So we know that the polyglycerol will be able to travel throughout the body for at least 25 hours until it reaches that acidic pH.
So our company is going to be more of a research and development entity. We're not looking to set up any manufacturing plants, so we're not going to need a ton of space to have big equipment or anything like that. Because of that, we're going to look to save costs from building our own space to rent space from the Pennovation Center, which is actually a part of the University of Pennsylvania in Philadelphia. Our rent would be $2,000 a month initially, and if we find later that we need more space, we can always rent additional space for more cost. So that's really nice that they have varying sizes depending on your need. Along with that rent, it includes 24/7 badge access so that we know our lab would be secure at all times and it wouldn't just have random students or other people wandering in. In addition, we have mail and package delivery services included, and the building is LEED Silver certified so that we know the water's clean, the air is going to be good, and we're not going to introduce contaminants into our materials that way. In addition, in Pennsylvania, there's a number of tax incentives that we'll be able to take advantage of to also cut down on our costs. They have a really nice research and development tax credit so that we'll actually be able to deduct up to 100% of all of our corporate net income tax whenever we do start turning a profit and things like that. And because we're renting, we're not going to have any income tax, not income tax, I'm sorry, property tax, like we would if we owned a piece of land. In the future, we'll also be able to take advantage of the job creation tax credit. Right now, you have to have a company with that creates 25 new jobs in order to take advantage of this credit. So when we grow in the future, we're hoping to be able to expand to that size.
So a little bit more about our synthesis process. This is the proposed structure of our synthesis. So it has two simultaneous processes, labeled 1 and 2, that will come together at the end. So we can go through each of these a little bit more individually. So for 1, we have 3 different syntheses, which you see in the dark purple, and those are outlined as 1A, 1B, and 1C, which we'll see closer in the next slide. So that includes a BBAG monomer, which is a Bach-protected butanolamine glycidyl ether monomer, and then different derivatives of that with the polyglycerol. So here are the chemicals that we would use for 1A, which is synthesizing that BBAG monomer. This will take approximately 24 hours to create, and it all happens at room temperature. And this on the right is what the BBAG monomer looks like, and on the far left is the glycidol ring. This is what we will use for 1B, which after the BBAG monomer, we add the polyglycerol by breaking open that ring and creating all of the generational groups to the terminal group. And then the last step in this process is deprotecting it so that BOC will actually come off and allow us to manipulate the terminal groups on the dendritic polyglycerol much easier. And then our second process is creating a doxorubicin derivative that will easily adhere to the end of the dendritic polyglycerol. So on the right is what we intend to attach to the doxorubicin. It's called a 6-mal derivative. I don't think any of us can actually pronounce it, so sorry about that. But so the process here is adding materials, including phosphate buffers and thiol binding doxorubicin, so that we have 2 final products, which we'll see a little bit down the line in the safety analysis.
So the space that we're renting provides us with things like benches and hoods. However, we are responsible for purchasing the equipment that we'll actually be doing the synthesis in. So that's mainly a round bottom flask with heaters and rotary evaporators. However, we'll still need to purchase things like pumps and chromatography columns. And to be able to test our process as we go along, we'll be using NMR and HPLC. We expect that to be able to cover the range, to be able to characterize our process as needed. However, towards the end, we will be outsourcing any final spectroscopy testing that needs to be done to Intertech Pharmaceuticals.
So for safety, some of our first chemicals that we use in the first couple steps of our process are fairly reactive. A lot of them are health hazards or flammable and are reactive with other chemicals. So in order to help counterbalance this, all of our raw materials will be stored in refrigerators in tightly closed containers separate from touching each other. We're going to install temperature sensors with alarms within our refrigerators, just so that if one happens to break, we're not going to be surprised and realize that it broke with an explosion. All of our reactions are going to be taking place at room temperature, so that will help with our chemicals not reaching their flash point. And in addition, to help minimize this risk, all of our lab coats will be anti-static and flame resistant, so that our lab workers won't have any risk to their personal well-being. Our final 2 products that we are producing are not reactive, not flammable, and they're not health hazards. So that's really nice that we're not going to have to worry about those too much. We are going to have to perform a stability analysis just to make sure that they stay how we want them for over a range of time, so that we know how they degrade, how they're should be stored, and stuff like that. So we performed a full FMEA and HAZUP analysis. This is only part of it to highlight the 2 most areas that are at risk. The first one is the condensation buildup within our refrigerator that I already talked about, where we'll minimize that with temperature sensors that will be checked biannually. The other one that could be a big issue is that if our rotary evaporator allows air into the system, this could cause a number of problems with the pressure and different things like that and chemicals escaping. So in order to counterbalance that, we're going to inspect the glass prior to every use and have good manufacturing processes with that. And we're going to have pressure sensors within this evaporator to make sure that it is pulling the proper vacuum. And we're also going to have biannual inspections on that.
So our timeline from conception to implementation is 7 years. We've split it up into 3 separate phases. The first phase being our identification and modeling and screening. So some of it was done with our research here. And then we're also in that phase accounting for any startups. So like getting all the equipment and hiring the workers that we would need. Phase 2 is expected to take the longest amount of time, about 5.5 years, and that's where we'll actually be doing the validation testing, testing for safety. And then phase 3 is our final sale. So our goal is to sell to an established biopharmaceutical company. So to do that, we'll need a patent, and then we'll also need to network with these different companies to be able to establish that sale. So a little breakdown of our organizational structure. The 4 of us will make up the members of the C-suite, and we'll have 3 main departments. So Mariah will be our CEO, and she'll be in charge of organizing everyone as well as doing part of that networking with the big companies. Isa will be in charge of finance and marketing, and we'll have 1 employee underneath him. And then Shannon will be responsible for the research and development team, where we'll have 3 lab techs that will help us with the process. And I will be responsible for human resources, and we'll have one part-time employee that will help us find the best talent out there.
So as Julie explained, the total number of employees that Dendroline will need is 9. And this table shows the annual salary for each employee. As you can see, the CEO will get $85,000, and CFO, CTO, CSO will all get $80,000 per year. And the plant accountant will receive $65,000. And each laboratory technician, which will need 3, will have $54,000, and the HR assistant will get $35,000 per year because he will be only working part-time in Dendroline. So the total cost come up to $587,000, and all of these salaries were determined by looking at similar salaries for similar positions in the city of Philadelphia, because that's where our location will be. And next we have the equipment specification and cost for the synthesis part. We will get several pumps, couple of rotary evaporators, and several round bottom flasks with different sizes, and several chromatography columns, and all of these will cost $21,000. And then we have the cost and specifications for the testing equipments. The NMR will cost us $900, and the high performance liquid chromatography will cost us $15,000. And then each sample that we will send to Intertech Pharmaceutical Service will cost us $100 for, as you can see in the bottom left, different testing that they have in the Intertech Pharmaceutical Service. And the total for those equipments will be $20,000. So for the overall cost summary, the personnel cost is $587,000. Again, the laboratory space and utilities, the utilities includes water, electricity, waste disposal, supplies. That's around $27,000, and then the patent and legal fees are around $50,000, and the raw material is around $360,000, while the equipment for synthesis and testing will be around $41,000. And you can see here how the cost distributes. Most of our expenses are for the personnel costs and the raw material costs, as you can see here. And so we all know that cancer is the second leading cause of death in the United States, and in 2016, the novel drug delivery system market in cancer was valued at $4.31 billion, and it is projected to reach $26.62 billion by 2025. And that is for some reasons. Reason one is because there is a rise in the number of research efforts into new therapies, and also an increase in the research budget for novel drug delivery. And also there's an increase of number of cancer diagnoses every year, and of course because of the growing public awareness through marketing. And also North America held the largest market share with 36.8% of the total market, and that is the largest market share by region. So So following the development and patenting, Dendroline will look to license its novel drug delivery system, and that with an establishment pharmaceutical company to get a full FDA approval and also to scale up our manufacturing. And in this table you can see the target companies that we are trying to reach and to partner and license for partnering and licensing.
So this field is heavily regulated by the FDA. So initially, we'll be working with them to do the designation request. So because it's categorized as a drug device combination product. So we'll do the initial steps with them, and then once we have the sale, the resulting purchasing company will take over and work with the in vitro testing and any other regulations, things that need to take place afterwards. But our safety is our priority, so we'll be implementing those things throughout. We'll also be working with the patent office to be able to do the steps that we need to get patented.
So at the end, our profit from sales is estimated to be $17 million, and the initial investment is going to be $9 million, so our net profit is going to be $8 million. And from that $8 million, we're going to use $1 million to reinvest in Dendroline. And the profit margin is around 20%, and that is based on historical data.
So in summary, we believe that the dendritic polymer can enhance the existing chemotherapy that's out there. So by having a business model that has low overhead and yields high profit, we expect this to be very successful. And at the end of the day, we hope to sell all intellectual property and then build a long-term partnership with a biopharmaceutical company. So I want to thank you for your time, and we'll take any questions.
Questions for Dendroline? For you,
Mine is just, of all of the places that you're thinking of partnering with, is there one that stands out in your mind as most likely to go for something like this?
I think Pfizer would probably be most likely. They actually have a venture capitalist section of their company that has a $50 million annual budget to invest in startup companies, where they can give $10 million per company. That's every year they have $50 million to invest in new companies. Normally they invest in companies that are in phase B of financing, which means they already have one round of investors and have progressed to a certain point where they're now looking for a second round. So we would have to do a little bit of startup with other investments before we approach them, I think, but they have a long history of partnering with smaller companies to help advance drug delivery systems.
Did one of you guys co-op for one of these? No, I thought that's somebody.
Someone did for
Co-ops.
Takeda,
Okay,
so that's
sort of, yeah, close to.
Yeah, they're working with Takeda. I don't think anybody else is doing anything on that scale.
Can I follow up? Who makes Doxorubicin?
There's a bunch of different companies that make it.
It's generic.
Yeah.
Yeah, I know you talked about partnerships and things like that. Are you aware of any like companies that are, have similar molecules or nanotechnology that they're working on to kind of get to the same point that you guys are trying to get to?
In our research, we haven't seen any other company that is trying to bring this to the market. There are a lot of university-specific researching sites that we got a lot of our data from, but I have not come across any data of them trying to market that product, just because I think they want to get further in their process. Like Mariah said, they want probably to be in phase 2 of their process before they can take that step.
So I had a couple of questions. So just, thinking about your idea, pH, using pH change to break a bond to release a drug is a great idea, right? Certainly, I think a lot of people have thought of doing that. But I wonder if you can think of some disadvantages of using a pH linker, pH-dependent linker for doxorubicin.
I think there are probably a few downsides, most being if your bond is broken up at like any specific pH. So if you're going through the body and you're in a neutral pH, and let's say like the sensitive linker that we choose specifically breaks at like 4.6, like that is going to be very difficult, I think, to withstand going throughout the body. But also, if we assume or calculate that a bond breaks at 4.6 and it does not, and it is closer to 7 and doesn't withstand all of kind of the complications within the body, I think that could be another unforeseen consequence.
Yeah, so I think, you know, in this kind of an approach, you have to be careful with the amount of doxorubicin that you would want to be released, and what, you know, where is that difference between being toxic to the body versus, you know, killing a cancer cell. Because the pH linker, usually they're kind of ranges, right? So you never really have an exact idea of, yes, we're gonna get 50 micrograms to release in that spot, right?
Right.
So I think that is an issue, and hopefully your company, the research would be done, and I know the FDA would ask for that kind of safety profile of proving how much of your doxorubicin is released at what pH. I think the other disadvantage of a pH-based linker is non-specific delivery. So it is true, right, obviously, that in cancers, you do have a more acidic environment, but you also have acidic environments in other parts of your body just naturally, right? And, you know, infection is one, and cancer patients often suffer from infection because they're on immunosuppressant drugs. So you could have a situation where, you know, you're actually releasing unintendedly doxorubicin in an infected area rather than in cancer. So somehow you have to figure out, if that happens, then, you know, the FDA would ask what's the consequence, right? But it's still a very unique, you know, idea, and I think where you would find a patentability is your whole molecule, right? You know, the linker that you're using, the polymer you're using, and then of course this pH sensitive release. The other general kind of question I had, and I'm not sure if you touched on this in your presentation, is why nano? You know, there's a lot of people who are against nano, right? They think that there's a lot of hype and there really isn't a lot of efficacy surrounding using nanoparticles in drug delivery. So convince those skeptics, you know, why do you want nano?
We focused on nano just because we read a bunch of different articles that said that certain range and size was better uptaken into the lymphatic system, and we thought that would be a good direct approach to get to cancer cells, whereas you said if it went through the bloodstream it might just reach random places that were infected and had a lower pH and then release it there. So if we focus on the lymphatic system, we thought it might be more likely to find the specific areas that we wanted to release the drug at, and so that nano range and sizes was best shown to follow that route.
Very good.
And just going off of that, so the final molecule that we showed up on the left that kind of looked like a fan, so there have been a few studies that actually put in a dye within the pH-sensitive linker so that it would release the doxorubicin and a recognizable color on whatever instrumentation they were using. So that would be another way, I think, to kind of not only talk to the FDA, but also the skeptics, so we could show them, like, this is where it's being released, and this is how much, and this is how we know.
Yeah, excellent. Those are good reasons for the skeptics. The other reason is that's where you can get something patented, right? There's a lot of patents. You guys maybe have seen this if you look at these bar graphs. You know, there's a lot of patents that are given out simply by making something nano, because a lot of the existing patents are around the micron regime, right? So it doesn't really have too much to do with science, but you know, in terms of forming a company and keeping it viable, having a nanoparticle allows you some potential patentability. So my last kind of general question is where the field is sort of going as well, in addition to having these, you know, kind of intelligent drug delivery molecules, is in developing molecules that can both detect whether you have cancer and then treat it with the same molecule. So do you know what that field is called by any chance? Where you're doing both a diagnostics and a therapy? So theranostics, right? It's actually, it should be the reverse, right? Because thera is therapy
Yeah.
and ostics is diagnostic, so it should somehow be reversed, but
Doesn't
nonetheless-
sound as good.
Right, right. I think you need to start with a thumb, right, to make it sound good. So I wonder, to pick your brains a little bit, how could you turn your molecule into a theranostic molecule? How could you do the therapy? I mean, the diagnostic part. You're already doing the therapy. How could you do the diagnostic part with your molecule? Any ideas?
Do you know?
And a great way to think about this is, how is cancer diagnosed today? What does a clinician do to determine if you have a tumor or cancer?
I'm not sure.
I don't think we did the research beginning
part.
from there, yeah.
And it actually could keep your company going, right? Like, you could sell your company based on the therapy and then have a phase 2 of your molecule to then explore after you have all this revenue that comes in from selling your company. So there's a lot of ways that you could incorporate other molecules into your polymer.
Mm-hmm.
So for example, if you incorporated a magnetic particle and you then had a receptor to go towards a cancer cell, that magnetic particle you can image using MRI, right, or other image techniques. So if your molecule then binds to a cancer cell and it has an element or a molecule in it that will show up in MRI, now the clinician can say, it looks like you have a concentrated number of cancer cells in that region. So, now maybe we want to activate it, maybe inject something that could make it more acidic, for example, to release doxorubicin to kill the cancer cells. So something just to think about, but definitely I believe in the next 5 years or so, we're going to see these molecules that can both detect whether you have cancer and then be activated to kill that tumor at the same time without 2 separate treatments, which is what we have today. Good.
Thank you.
Thank you.
Excellent.